up to talk about how the tRNA has the amino acids on them. The thing that glues the tRNA and the amino acids together are an enzyme or enzymes called tRNA synthases. And they join the tRNA with the amino acids that the mRNA code for. And they come together as a group. Let's talk just a little bit about rRNA then. rRNA, they are called rRNA for ribosomal RNA. All the RNAs are produced from DNA. Anything in our body is produced from the instructions in DNA. They are part of a complex of a whole bunch of proteins along with the rRNA to make up the ribosome. So they're just one part of the ribosome. Okay, so here's a little bit about the ribosome. The ribosome is actually made of two parts. It's made of a large subunit portion and a small subunit. The ribosomes are not one complete part because they wouldn't fit out of the nucleus then, where they are manufactured. The larger subunit has two binding sites where the tRNAs go, and then they have what's called a catalytic site. So whatever amino acid the tRNA brings over that the mRNA here on the small subunit codes for, those two amino acids go through a dehydration synthesis. Remember way back? They make a bond between the two amino acids to form a peptide bond between them. And then those peptide bonds add two amino acids together, and then they move over one. A new tRNA comes over with the amino acid, and then another peptide bond is formed. And every time you add a new amino acid, another peptide bond is formed until you get the complete protein. This whole process terminates, meaning the translation process terminates at that terminator sequence, and then the entire new protein gets lifted off of that last catalytic site, or excuse me, the last binding site of the large subunit, and the protein will go wherever it's needed in the cell. Ribosomes can be found in two places in the cytoplasm. One is they can be floating free in the cytoplasm, and the other place is they can be found on the rough endoplasmic reticulum. The signal sequence that is found near the start codon of the DNA instructions, which is then again translated into an mRNA transcript, knows where to go because of the signal sequence on the endoplasmic reticulum. Almost all protein sequences have this signal sequence that says, this is where I'm going, this is where I'm going to be used in the cell, and it has that information. So let's talk about a little more detail about the complete process of translation. So the first step is called chain initiation. It starts at the start codon. The start codon is a base triplet called AUG. Codes for methionine. It says we're going to start our protein sequence here. The tRNA comes over, and it brings over that methionine on its backpack to this mRNA codon. The tRNA is called the anticodon. And then the large and small ribosomal subunits come together. So everybody's kind of like coming together at once. They're all merging to begin this process of chain initiation of translation. Then you have chain elongation. So the methionine is red, the AUG codes for the tRNA anticodon to bring over methionine, and then the next tRNA brings over whatever the next mRNA codon codes for, and you just keep adding and keep adding those peptide bonds, and everybody moves over one, and it keeps going until you have a chain of amino acids. Then you get to the stop codon. Terminator sequence says, all right, we're done. We've completed it. Remember, there's 3 possible stop codons and the 64 possible codons. Then everything starts to separate. The tRNA goes off to get some new amino acids glued on by tRNA synthases. The mRNA breaks up. The large and small ribosomal subunits break up. So everything's done, released. The protein goes wherever it needs to go in the cell. All right, so that's kind of the basics of it. So let's take a look at starting from DNA here and going down to making our protein sequence. So remember that in the nucleus, this top part here, the DNA is double-sided, and one side of the DNA is the template strand, which is going to be copied into a complementary mRNA. So you will know on the exam, if I say this DNA is double-sided, I will tell you that is the template strand. I will tell you which side it is. The template strand gets complementary mRNA transcript. So the T is going to complement A, the A is going to complement U, C gets complementary GGG, T complements A, C complements G, and those two A's get complements of U. The mRNA is going to leave the nucleus, it's going to go out into the cytoplasm, it's going to attach to a large ribosomal subunit, small ribosomal subunit comes over, and tRNA comes over to give the complement of the amino acid that the codons, the mRNA, every 3 of them, every 3 nucleotides of the mRNA, whatever they code for, tRNA brings over that amino acid. So AUG is a start codon. It gets a complementary U, A, C, tRNA anticodon, which is carrying methionine that AUG codes for. So again, remember. The mRNA is what codes for the amino acid. The anticodon tRNA is only bringing it over. mRNA AUG, when you look that up in the codon chart, you find methionine. You do not look up UAC. You don't look up the tRNA. You always look up the mRNA. So mRNA we look up to find what tRNA is bringing over. mRNA codes for amino acids. Make sure you remember that. The next codons are GGA. GGA, code for glycine, and the GUU, code for valine. So let's take a look at this in pictures another way. So initiation means that there's a small ribosomal subunit. It has the anticodon to AUG. The anticodon to AUG is UAC, which carries the amino acid that AUG codes for, which is methionine. A small subunit has UAC carrying methionine. AUG comes over and pairs up with the anticodon. The next thing that's going to happen is the large ribosomal subunit is going to join up. Large, small ribosomal subunits are joined up. The AUG start codon has its methionine. Next, what's going to happen is this second binding site is going to call over the appropriate anticodon for GUU. GUU gets the anticodon CAA. GUU codes for valine. At the catalytic site, a peptide bond, a dehydration synthesis between these two amino acids is going to occur. They'll get their peptide bond, and then everybody's going to move over one. The next binding site, you have CAU. The appropriate anticodon brings over GUA. The CAU codes for the amino acid histine. At the catalytic site, valine and histine are going to undergo a dehydration synthesis to make a peptide bond. And then everyone's going to move over another one. This keeps happening, everybody keeps moving over until the entire protein is made. Once that is made, the stop codon, terminator sequence are going to say, everybody's done. So the mRNA can leave, the tRNAs can go off, find tRNA synthase to get new amino acid backpacks. The large and small subunits dissociate, they come apart, and the protein goes where it's needed in the cell. So, a few questions. Imagine that the template strand of a gene has the sequence of TAC. So that means the DNA template strand. It says what sequence of anticodon would code for this? So remember, you're going, DNA template is going to get a complementary mRNA. DNA template strand will get a complementary mRNA. Those are the codons. The mRNA will leave the nucleus and will go out and get paired up with the tRNA anticodons. So in this question, you have to know a couple of things, right? You have to know that the anticodons are the tRNA. So let's make our mRNA complementary codons. So we're going to make our complement. T is going to complement A. We're going from DNA to RNA. A will complement U. C will complement G. There's that AUG, right? That start codon. And we're going to have a complement of AUG. A complements U. We don't use T's anymore because we are still going RNA to RNA. U complements A, and G complements C. So just to give you an idea of how to conquer this question. So it says ATG because the anticodon is complementary to the template strand. No, right? AUG because the anticodon is complementary to the template strand. Not the anticodon, the codon, right? UAC, because the anticodon has the same sequence as the template strand, but has a U instead of a T. Look at that. So there's a couple ways you can, I always like to just go and do the complementary mRNA and do the complementary tRNA. You could just go, okay, this is going to be, the template strand of the DNA is going to be the same thing as the anticodons, except where you have a T, you'll replace that with a U. So you have TAC, here you'll have UAC. Almost the same, except for the T is replaced by a U when you're looking at the anticodon. So there's a couple of ways you can look at it. So that looks good, number 3. And then TAC, because the anticodon has the same sequence as the template strand, not the same. There's a U instead of a T. Anybody? Questions on that? All right, so general qualities about the genetic code. It's universal for all organisms. This is, going back to a common ancestor, we do see that all organisms have A's and T's and G's and C's and U's, and so we do see that universally. It's based on 64 codons. Comes with a start codon and stop codons. And the redundancy means that a mutation can be made and it will still code for the same amino acid, potentially. Sometimes, not always, but at least we have a little bit of wiggle room there with mutations. And so again, remember when we're talking about this redundancy, and what I mean by that is that, for example, if we look at leucine, leucine can have a bunch of mutations here, that if you have, let's say, for example, you're supposed to have CUU, but instead you get CUC, you have that mutation in the third amino acid, the third nucleotide in the codon, they both code for leucine. So mistakes can be made, but they can still have the outcome of the same amino acid. So that redundancy, it helps to limit the outcomes of mutations. All right, mutations have different effects on protein structure. So let's take a look at some examples as well. So this is what I mean by the new codon may specify the same amino acid sequence. The protein's unchanged. UUU, you have a mutation in that last U to a C, but both of them code for phenylalanine, so no big deal, mutation. New protein may end up being equivalent, same thing, because those mutations and that redundancy code for the same thing. Here's the bad thing. The mutation, it's so messy that you get a completely different protein. Or the protein, it doesn't even equate to a protein, it just comes something like a blob of amino acids and does nothing for the cell. So, for example, let's say you have UUU, AAA, CCC, and GGG, which, if this is the mRNA, codes for phenylalanine, UUU, AAA, lysine, CCC, proline, and GGG, glycine. Let's say that there's a deletion mutation, that one of the U's is accidentally deleted out, so everything switches up or shifts one over. So instead of UUU, you have UUA, which is supposed to code for phenylalanine, but now it codes for lysine. So that's something different. And then you have AAC. AAC codes for asparagine, but we were supposed to have lysine. And then the next one, CCG. Look at that though. We have proline, and we end up with another proline. So sometimes they'll be different, but sometimes they'll be the same. Proteins are really, really long. They're long chains of amino acids, so the likelihood of this equating to the same functional protein, probably not. Especially if the mutation is very early on in the mRNA template. The mRNA transcript. All right, so how common are mutations? 10 to the 5th, 10 to the 6th, eggs or sperm may carry a significant mutation. So it's not very common. Most mutations do one of three things. They're either harmful, neutral, or they create a new phenotype. They might create a new option that's not necessarily bad. It might be beneficial. So sometimes mutations can lead to a beneficial outcome. Those that are beneficial may spread faster through a population than those that are not beneficial. So last thing, we're going to do a few practice problems. All right, so if the DNA triplets were GAT-TAC, what would the mRNA codons be? So we have GAT. What would be complementary to that? Let's go G complements C. So even if you just start there, here we're looking at, we know C is the complement, right? So we can get rid of 1 and 4 right away. G complements C. A complements T or U? U, right, okay. So we know there's no T's in our mRNAs, so we can get rid of 2. You don't even have to go much further. I would just make sure the rest of it matches. G complements C, A complements U, T complements A, T complements A, C complements G, and A complements U. So yes, 3 is definitely the best answer. All right, now if we have a segment of the DNA that has a sequence of that, the messenger RNA complementary to it would have what sequence? So again, I'm going to start out looking at C complements G. Nope, nope, nope. Okay, now it's the only one that has a G, so I'm going to check out the rest of number 4. C complements G, G complements C, A complements U, T complements A, G complements C, C complements G, T complements A complements U, and C complements G. Oh, sorry. I could have eliminated that one anyway. Okay, good. Okay, the DNA triplets are that, the tRNA anticodons. So remember, you're taking this one step further. Remember, there's a couple ways to look at this. You could go from the DNA template to the mRNA and then get your tRNA. Or your anticodons will be the same thing as the DNA template, except for anywhere you have T, you will have U. So we do this way. So if I have my DNA template, the A will complement U, A complements U, G complements C, C complements G, A complements U, and T complements A. That's the mRNA codons. To make my tRNA anticodons, I'm going to take the complement of the mRNA codons, so that U complements A, U complements A, C complements G, G complements C, U complements A, and A complements U. So these two are similar, except for in the DNA, you have thymines or T's, And in the RNA, the anticodon tRNA, you have U's where you would have T's in the template strand.